So welcome to today's HIRN webinar, the Human Islet Research Network. This one is going to be on a library of diabetes resources that provides opportunities and applications for your lab. I'm Joyce Niland. I'm the director of the Coordinating Center, a co-director with John Kaddis, and we're located at City of Hope. And today's presenters are going to include Dr. Kaddis, Dr. Sarah Tersey from the University of Chicago, and Dr. Eddie James from Benaroya Research Institute. So to tell you just a little bit about the Human Islet Research Network, for those of you less familiar, this network has been in existence since 2014 and now consists of 5 consortia and the Coordinating Center, which we call the HIRN, the Human Islet Research Enhancement Center. The consortia include, over to the left, the consortium on beta cell death and survival, then the consortium on modeling autoimmune interactions, or CMI. CHIB is the consortium on human islet biomimetics. CTAR is the consortium on targeting and regeneration. And the most recent consortium to join is the HPAC, the Human Pancreas Analysis Consortium. Together, they're studying how beta cells are lost or damaged in type 1 diabetes, how they can be replaced, how it can be prevented. There are over 200 investigators at 85 institutions across 9 countries that are currently involved in the HIRN. And more are being added as new opportunities for funding become available. You should keep an eye out for that. And if you want to learn more about the network, we have published, or in the process of publishing, a review series in molecular metabolism. Here's the first article that the Trans Network Committee put together to give an overview, and then there's about 4 more articles published already and another 4 in progress. So that's another way to learn about the HIRN. And another way to find out more is our HIRN website. We most recently just published our Year 5, Year 6 Executive Summary Report. It's a nice synthesis and digestion of all the research that's going on, so take a look at that. It's published out on the HIRN website, which is hirnnetwork.org with just 1 N. And what we're going to talk about today will be recorded and posted on YouTube, as have all of our past tutorials and webinars. So you can always go out there and look at the videos if you miss one or would like to review anything. Tell your colleagues about that as well as a resource. And that's what HIRN is all about, is to provide networking and resources and technology for the field. And so today we're going to talk to you about something that our HIRN has developed, which is the HIRN resource browser. It's got a number of different activities and resources out there. And the HIRN is composed of myself and John Kaddis as the co-principal investigators. Layla Rouse is our program manager. Sandy Bashir is the research data curator who has helped to curate the many resources you'll hear about today. And Dave Coe is the technical lead for the development of this website, with assistance from Nellie Berger as the project assistant. So there are many, many entries on this website, over 1,900 in the various categories that you see here. And John is going to give you an overview of this, and then we're going to have real-life examples from Sarah and Eddie about utilizing these resources and contributing to them. And in developing the HIRN resource browser, we have adopted and helped promote the key NIH principle you may have heard of FAIR, or fairness. And this stands for resources that are findable. They need to be accessible, interoperable, and reusable. And so we're trying to adopt all of these principles as we promote the resources that are on the browser. And this is something that you all, of course, need to adopt as well and write about in your grants. So by utilizing the resource browser, it'll help you with these principles as well. So let's go to the discussion and topics and reminders for today. We're going to discuss first an overview of the HIRN resource browser by John Kaddis. And then Sarah Tersey is going to talk about some tools and data sets useful for studying T1D all the way from mouse to human models. Then Eddie James will look at highlighting resources to enable antigen-specific studies. Just a few reminders for today. As I said, the presentation will be recorded and posted on YouTube. So please mute your audio and turn off your personal cameras during the presentation, and we'll be sure to save time for discussion at the end when you can raise your hand and unmute or use the chat feature to ask any questions as well. So with that, I'm going to stop sharing and turn it over to John for the overview of the HIRN resource browser. John?
Yep. Thank you very much, Joyce. I appreciate it. Okay. Hopefully everyone can see that. And I'm going to pick up where Joyce left off, our website. And in particular, I want to highlight one element of the website where we list the resources available. And I briefly want to touch on that. So it's true, we are going to go over the resource browser today, but there are several other things that you may or may not be aware of. First is the PancDB. And as Joyce mentioned, this is a resource from the Human Pancreas Analysis Program. And actually just last week, if you joined us or if you have been joining us for these webinar series, you will note that they went over beautifully the work that's been done and the updates that's been done there. So you can always visit the webinar there. I think there's probably something like over 30,000 data files in there. So it's a really nice resource. Another one is the Integrated Islet Distribution Program, and Dr. Joyce Niland is the PI of that program. And essentially, you can get fresh islets, you can get frozen islets, you can get them from type 2. You might even be able to get them from type 1 on very rare occasion, but there's also a research data repository. So it's not just islets, you can actually get the data on these donors as well. So that's another resource. Most of us are probably aware of NPOD, where not only can we get pancreas tissue, but we can get spleen or duodenum or even other tissues that are coming online as well. And we can get them from different sections in the pancreas. So that's really a beautiful resource that's been helpful to many in HIRN, and I'm sure to you as well that are tuning in. There's another resource you may or may not be aware of, and that's called the Pancreas Atlas. And that essentially currently has 6 collections that are cataloging different images. So lots of different data available in image form. And one day we might be lucky and we'll be able to get them to come speak in one of these webinars. And then as Joyce mentioned, we've got the webinar series and we're cataloging all that. Okay, so with that, let's jump into the resource browser. Joyce has shown you what it looks like. And I thought the best way to move through this would be to illustrate a couple of use cases to get you familiar with that. So you're an investigator and your question is today, I need a polyclonal antibody that targets insulin. What do you have? So you'd click on the antibodies and you'd come to this page. And if you notice on the left-hand panel, you can click through a series of search features. And when you do, you get these results. Now we'll choose one of those results, which would be this one I've selected for you here. And then we can go and we can look at this. So what do we get when we look at this? So you actually get the antibody, you get where it is manufactured or what vendor it's available to. But probably what would be more useful is if you know, has this antibody been used by anybody? And if so, what applications has it been used in? So later or down the page, depending on the length of the page, you'll find this box to the lower left, and you'll see what application the resource has actually been used at, in this case, for an immunofluorescence assay. But what you might not realize is that you can click on what I've highlighted for you here, and if someone has taken the time to share that data, you can actually pull up the dilution series. In this case, this is the data that Dr. Chris Wright's lab has shared with us from Vanderbilt. So he's went through for a number of the insulin antibodies this being key to a lot of us in the field. And he's shared a lot of his data. So we're very thankful to him. But you can actually go and get the data and see if it's worked well or hasn't worked well. But of course, this isn't the only, antibody that's there. You might need another antibody. And so you can go into the search page, and we're accustomed to Googling. So you can type in what you're looking for, and then you can get it. So that's one quick way of going through what our biggest category, which is the antibody. But what we also do is, coming back to the homepage, is we highlight different collections that might be of particular interest. I did mention the Chris Wright collection, where he has actually tested a number of these antibodies, but there are other special collections in which investigators have volunteered their time, their effort to share different elements of the data. Today, you'll hear one of them from Dr. Eddie James, where he's gone through and made available a lot of tetramers, the box that you see down there on the left. But let's go to our second biggest category, which is data sets. And let's ask a question. How many mouse-related data sets are available? So again, let's click on that box and let's see what we find. We go through the search box. We can find that there are 64 data sets that relate to mice. We can choose one of those data sets. And it looks rather plain, but you, again, might not realize that you can actually click on the identifier and it will take you to the geo entry where you can obtain and pull that data. You can actually also go to the paper and pull that, and you might expect that, so that's fine. If you're programmatically inclined, that means if you're a bioinformatician, or if you're just familiar with data and know how to use things called application programming interfaces, there's two additional ways that you can pull the data today. Now, we realize that this isn't user-friendly for the everyday biologist, so we're working on ways to give you direct access to this. But I just wanted to briefly point out that you can now go in and download every single data set, not just a mouse-related data set. So all the particular data sets and where they're located and the type of data set it is, you can actually do that. But we are the Human Islet Research Network. So you might ask, well, I want RNA-seq data sets, and I want those only from human samples. So again, do the filtering on the left, get the data that you want on the right. So you can have that available to you, and then you can go and pull that together, and maybe you're interested in some sort of meta-analysis or for some other purpose. Now, again, back to the home page, what we also have, if you scroll down to the bottom of the home page, is we have our newest resources. So we're adding resources every day or every other day. And so you can always get to those here as well. So you can, those will get highlighted for you. But let's jump into our third category. And this is the final example that I'll run through with you. And that is model organisms. So your question might be, is there a mouse for the gene that I'm studying? And again, going through the filtering criteria, trying to locate it. Here I just selected one. And the only thing I really want to point out here that hasn't been pointed out is this, the thing at the very bottom here where it says related resources, because in the process of curation, what we also try to do is we try to link up a particular antibody, or in this case, a mouse, with other resources that might have been used in that same experiment. So in other words, if a paper was published and it reported this mouse and an antibody and a data set, we try to link those things together if we can. And then we make that available to you here as a window to what other resources might be related or linked to the one that you're interested in. But what do you do if you can't find the model, a model that you're interested in, and the resource browser just doesn't have it for you? In that case, what we do is we send your search results to a collaborator called the NIDDK Information Network. And there's a reason why we do that, a few reasons why. It's true, they are a collaborator and they help us with registering data, getting IDs on the data, and as Joyce mentioned, making data fair. But their repository has over 800 million different resources. So if we don't have it for you here, DKNet would likely or hope to have it for you there. Okay, so with that, I just want to summarize what I've told you. There are lots of different databases and repositories available to you. You can go to our website and get them. We're curating different tools and reagents and other things that you might find useful in your experiments day to day. And there isn't any special permissions required. You can always access this. And we also try to link out the names of the people that you can contact in order to ask for a particular resource if it isn't publicly available. So with that, I would like to ask that Dr. Sarah Tersey start sharing her slides, and she's going to be the first one to share with you applications from resources in the in this repository. Dr. Tersey is an islet biologist. She is at the University of Chicago. She's a research associate professor there, and she's been in the field for a number of years. And what's been developing is a very interesting story and a very recent publication that she's going to share with us today. Thank you, Dr. Tersey.
Thank you, John. And hopefully you can see that okay?
Yes.
Great. All right, so today, like John mentioned, I'm going to talk about one of our recent publications, and then also intersperse that with some of these databases that we use, some of the resources that we used, and some that we have now provided to that data set. And so for my talk, we're going to be talking about pro-inflammatory signaling in beta cells and how that triggers autoimmunity. During type 1 diabetes, beta cell mass and function are diminished due to autoimmunity, leading to insufficient insulin secretion and hyperglycemia. Our lab has focused on the role of the beta cell during this time. The current hypothesis suggests that beta cells first get exposed to the unknown environmental trigger, which leads to inflammation. The beta cells then exhibit activation of the unfolded protein response, followed by ER stress, oxidative stress, beta cell dysfunction, and ultimately beta cell death. This results then in the release of antigens and neoantigens, which leads to immune evasion and further propagates the cycle. And it cycles around, and you have this autoimmunity and loss of beta cells, leading to type 1 diabetes. So we have previously shown, there's a lot of papers out there, that 12-15 locks, otherwise known as 12 locks in humans, you'll hear me refer to as both, is increased during inflammation. And here, as one example, is some tissue samples that were obtained from NPOD, which John mentioned. And so we have non-diabetic individuals and very, very low expression of 12 locks here in red in beta cells, as shown here in insulin. But when individuals with type 1 diabetes are looked at, you see this nice increase of 12 locks, and that's also persistent in autoantibody positive individuals. So in pancreas and beta cells expressing inflammation, we see this high increase of 12-15 locks. So what 12-15 locks does is, in times of inflammation, this enzyme is activated. It breaks down arachidonic acid, which has been released from the membranes, into a pro-inflammatory mediator called 12-HEAT. And 12-HEAT has been shown by us and several other people that it is involved in the production of chemokines, generation of oxidative stress, and it promotes a pro-inflammatory state in general within the islet, as well as within immune cells. And so here, looking at just 12-HEAT serum levels have been shown to be increased also during this time. So in newly diagnosed individuals with type 1 diabetes, there's this nice increase of 12-HEAT in the serum, which is probably not just coming from beta cells, but other cells, but it is coming from beta cells as well. And so our lab, like I mentioned in the beginning, is interested in further understanding the role that the beta cell itself plays in propagating its own demise and autoimmunity. And basically, how do we get from this nice, healthy looking islet to an islet that is inflamed and inviting immune cells to destroy it, right? So in order to start to answer that question, we have developed a beta-specific knockdown mouse. So here we have the ALOX15, which is the 1215 LOX gene in the mouse that's been floxed. And we crossed that to the PBX-PB Cree ERT mouse, which is expressed upon tamoxifen injection in beta cells specifically. And we back-crossed both those mice onto the NOD mouse, which is a model of type 1 diabetes. It develops autoimmune and invasion of the beta cells. And the mice develop diabetes at around between 12 and 25 weeks of age. In a manner similar to humans. It's as close model as we have right now. And the HIRN has been involved in developing humanized models of these guys. But this is the one right now that we are using. And so what we did is we back-crossed onto the NOD background, and then we crossed those two mice together. So that upon tamoxifen injection, which we did at 6 weeks of age, we developed the beta cell specific knockout mouse. Now, John mentioned the beta database and the model and organisms. So both these two mice have been submitted to Jackson and to the database website. So you can find that information there without having to come to us. They will be available at Jackson, and specifically the Cree line is a beta cell specific driver. It would be great for use in studying islets and type 1 diabetes. And so here, just to show, we see a nice 80 to 90% reduction of our gene of interest when we use this line. So really quick, I'm going to take a quick segue and show you guys the paper that we published in 2021. We also looked at 12-lipogenase, but we looked at this in myeloid cells. So what we did is the same thing. We use the same mice, ALOX15 mice, but we also have this lysem Cree mouse that we have back-crossed onto the NOD mouse. From development, so and it's in the myeloid specific cell knockout. So again, we created myeloid cell mice, and those mice are also, if not in the database, they will be, and they're available from Jackson. And so those mice, in the Cree mice here, so there's a lysem, right? The myeloid cell specific knockout, or Cree driver, become nicely diabetic at about an 75 to 80% rate, and our knockouts, when we knock out ALOX15 and the macrophages are resistant to that diabetes development. So segueing back, when we knock out ALOX15, specifically in the beta cells of females, we see a nice protection. So here, this is diabetes incidence, and as the mice become diabetic, they fall in the number of percentage here. So this one here in blue on the left-hand side is the Cree positive mice. So these mice both have tamoxifen and the Cree expressor and still develop diabetes nicely. Over on the right-hand side are other controls. So we have the flox control as well as the heterozygous knockout control, and they both develop diabetes as well. So this is female mice, and then the NOD mice, females develop diabetes with a much greater incidence than males. You can see our knockouts here in pink that are nicely protected. In our males, our Cree mice actually very nicely develop diabetes with about 50% rate. It's normal, on average, they develop anywhere from 10 to 50%. And in our colony, we have really nice diabetes incidence to our males here. The male knockouts remained completely protected from developing diabetes over the course of their lifetime. And then here, of course, here is our controls as well. All the rest of the controls that I use and the mice that I talk about will be females because they're more likely to develop diabetes, and we're looking at stages before. So we want a greater percentage of mice that have developed diabetes. And my actual control mice are the Cree positive mice and not the flox and the heterozygous, because the Crees can influence diabetes. But here we've shown that that is not the case with any of our controls. So first, to see what was happening and why we have this protection from diabetes incidence, we looked at the pancreas, and we see a much greater percentage of insulin positive beta cells at 12 weeks of age. So at 8 weeks, which is only a week, 2 weeks after we induce gene deletion, there's not much going on, but by 12 weeks, they have this preservation of beta cell mass. So they're not really gaining beta cell mass, but they have preserved it from dying off that we see with the control mice. And in doing so, what we used, John nicely noted it, the contribution by Chris Wright's group. We participated in some of these, and we studied all these different insulin antibodies. It's one of the things that there's good ones and there's bad ones. And so here, instead of testing them all in your lab, you can go straight to the website and the database and find which ones that you want to use. It's a great resource, and I highly recommend it for anybody that's new to doing insulin staining. And then we also looked at encephalitis and the amount of immune cells coming into the beta cell. And again, here's some antibodies that we used, and they've all been submitted to the database, and you can find them, so you don't have to find them for yourself. And what you can see is that at the early stage, at 8 weeks, right after the deletion, there's not much going on. But by 12 weeks, there's this nice reduction of encephalitis, and these islets start to appear healthy, and they've sort of blocked that autoimmunity from coming in. Looking at that 12-week stage, we want to know what was the makeup of the ones that were there and what was going on. And so we're looking at flow in the pancreatic lymph nodes, so the immune cells that are coming into the pancreas. And again, these flow antibodies have been added, and we see a reduction of all immune cells. So both pro-inflammatory and anti-inflammatory in general, just immune cells are lower and have been suppressed, and the pancreas is allowed to stay healthy in that sense. But to really understand what role the beta cell is playing, right? We knocked down one gene in the beta cell, and how is that influencing it? We went to an early stage. And so we went to 8 weeks right after deletion, and we started to look at what's going on in the pancreas and the beta cell. So one of the things that we did is single cell RNA sequencing of the islets. And in doing so, we could look at the immune cells that are next to the islets. We can look at the beta cells and the alpha cells and everything else that's going on. And we compared the positive mice with our beta cell specific knockout, and we started to analyze and see what's there. On John's slides, you can see our data set. It was right there on the bottom. That has been added here. It can link you to Geo, and you can pull it out yourself. And so not only does it have our gene of interest, but it does have single cell sequencing from NOD mice that you could then compare this to CD1 mice or CD7 black snake mice and see what's different, and look at the different immune profiles and stuff like that, which might be of interest to some people. So that's there, it's been uploaded, and you can see that. And so what we found when we looked at the islets themselves, the beta cells, right? So we had 5 clusters of beta cells. And if we look specifically at the beta cells, what we saw were changes that enhanced cell function and survival, and were related to the antioxidant response. So those are things that we knew that ALOX15 was responsible for, or we have seen changes in before, and now we can see that here in these mice, specifically antioxidant response genes. But what we found that was unique and interesting was that in one of the cell clusters, called the beta cell cluster 5, is this little one right here highlighted in red, you can see, was an upregulation of CD274. The CD274 is the gene for PDL1. And we not only saw an increased expression of CD224, we also saw an increased number of those cells in the cell cluster 5 from our knockout mice. And then we confirmed this with CyTOF, which I will talk about later. So just protein expression in the pancreas, PDL1 levels were higher in endocrine cells, so likely in islets, but specifically in this beta cell cluster 5. And so PDL1 is an immune checkpoint protein that's expressed on the cell surface and mediates peripheral T cell tolerance and autoimmunity via interaction with its receptor PD-1. So when you have more PD-1 and PDL1, you get a greater increase of Treg development and a suppression of T effector cells. And when you have less PD-1, you get a higher level of the pro-inflammatory effector cells than you see with the T regulatory cells. And so this was of great interest to us, right? Because if we can make more Treg cells, that would be awesome. And so we continued and we looked at PD-1 protein levels and to see them actually in the beta cells. And you see very nicely here, the PDL1 levels are increased in the insulin positive cells and with quantitation over here. And this is, again, just another highlighted use of the databases. And here's the anti-PDL1 antibody. We got it from AbCam. There's the vendor number. And then, like John showed, you can see applications that it was used for as well. So also using single cell RNA sequencing, we looked at T cell genes. And if you saw, remember at 12 weeks, there was tons of changes to the T cells and effector cells. But at 8 weeks, what we saw, both by flow and single cell RNA-seq, was an upregulation of anti-inflammatory T cell genes. So genes that are going to lead to T regulatory cells. And we saw this increase. So this is in gene expression. And we also looked at protein expression. So we use mass cytometry, called cytometry time of flight, or CyTOF. And we did this both in pancreatic lymph nodes and in the pancreas itself. And what we really saw were no changes except in regulatory T cells. And this is shown here, right here. You see a nice increase of CD25 positive, which cells, which is the one CyTOF marker for regulatory T cells. We did see a slight reduction in myeloid cell, inflammatory myeloid cells as well, shown here. And then we confirmed our T regulatory cells by flow cytometry. And you can see here, this nice FOXP3 was increased. So at 8 weeks of age, the only thing that we're really seeing is an increase of T regulatory cells. And then by 12 weeks, all those have diminished, all the immune cells coming in to the pancreas. And so one of the other interesting things we found in single cell RNA-seq was knocking out 12 blocks just in the beta cell also increased not only PD-L1 in the islet and the beta cells themselves, but also PD-L1 was increased in macrophages and dendritic cell clusters. So here's the macrophages and the dendritic cells, and you can see this nice increase as here as well. And then we confirmed that again by CyTOF. So this is myeloid cells, and then dendritic cells, and then macrophages all had increased PD-L1 levels. And so next we did, we wanted to see if PD-L1 was truly affecting these mice. So we treated our knockout mice first with tamoxifen to knock out the gene, right? And we followed that up with anti-PD-L1. So an antibody against PD-L1 to block the protein that was there. R is control, which is the anti-IgG antibody treatment. And then we followed them for 1 week and did some analysis on them. And what we saw was that the anti-PD-L1, or mice that received anti-PD-L1, had an increase of blood glucose within 7 days. By 9 days, and they're already overtly diabetic, which if you remember, NOD mice don't become diabetic at this stage in general, but our beta cell specific knockout mice definitely do not. And what we saw is an increase of insulitis, as well as a drastic reduction of beta cell mass. And these islets here are really the only ones that we saw in all of the mice. So mice is great, they're wonderful, useful tools, and we can't really go and knock out ALOX15 in human samples, but this is the Human Islet Consortia, and we want to translate our research. So we next took human islets, which we received from both the IIDP, which John talked about and Joyce talked about, as well as from the University of Alberta's Islet Core. And we treated them with either cytokines to induce an inflammatory state, or cytokines plus ML355, which is an inhibitor to the human version of 12 blocks. And you can see here that with cytokines, you see an increase of PD-L1, probably as a compensatory mechanism, and that is known and been published. But when we treat with ML355, we see an even greater increase of PD-L1, consistent with our mouse data. And there's some more antibodies there on that website that are human specific that you can find as well. And then to move a little bit further, one of the data sets that's in the HIRN database we pulled from, it's with our collaborator, some of our collaborators at Indiana University, and we did some work and we reanalyzed that data set. So this is deep sequencing of human islets that were treated with and without cytokines. And then this time around, what we did in our reanalysis was look at alternatively spliced RNA transcripts. And what we found were pathways that were related to protein transport, processing, and antigen presentation, which is consistent with alterations in mRNA translational processes in beta cells and with the activation of the integrated stress response. So the integrated stress response is made up several different signals that can come in, and they phosphorylate eIF2 alpha. And the phosphorylation of the eIF2 alpha leads to several different things. The first is complete global translational block, stress granule formation, and then the activation of the ATF4, which increases the transcription of unfolded protein response target genes. And so we then went back and looked, used the human islets that we used before, and we treated with a blocker of the integrated stress response. And what you see here is that when we treat with cytokines followed by ISRA, which is an inhibitor, you see the same sort of increase of PD-L1 that you did with the ML355. And so we asked the question, is our 12-LOX pathway related to the integrated stress response? And so we looked at using more antibodies, right, that we can find on the database site, that markers of the integrated stress response. So first, EIF, phosphorylated eIF2 alpha was decreased, right? So we have a reduction in one of the first signals of the integrated stress response. And then we also saw a decrease of G3BP1, which marks stress granule formation. We have a very nice decrease here. So suggesting that in response to our deletion of 12-LOX in the beta cells, we're seeing a decrease of the integrated stress response in these mice altogether, right? And so, like I mentioned in the very beginning, we're trying to answer the question, what is the contribution of the beta cell to pathogenesis of type 1 diabetes? And here in this paper, we knocked out very specifically one gene in only the beta cells. And we saw that by doing that, we could prevent this from happening, right? So we still had beta cell death because we were knocking it out at a later stage, but we were able to block the immune cells from coming in by altering just one gene in the beta cell. And so we think, you know, the beta cell is really playing this great big contribution to autoimmunity, and perhaps that's through the integrated stress response, which is what then our HIRN grant is looking for, and we're doing more research on there. With that, I would like to thank our lab. So I'm a member of Regular Benaroya's lab at the University of Chicago. Annie Pineras, who was a postdoc while we were at Indiana University, was the primary author on this paper. And then several other members of our lab have worked on this, Abhishek, Faye, and Andy. And then we have collaborators from both IU as well as others that provided mouse models and helped us along the way. We have funding from HIRN, NIH, but we got islets from the IIDP as well as the University of Alberta.
Thank you very much, Sarah, for taking us through step by step this new work that you've done. We're going to hold questions until the end. If you have anything that you'd like to put in the chat, please go ahead and do so. But I'm going to ask Dr. Eddie James to share his screen and queue up for the next presentation, where he's going to talk about tetramers. So we're going to switch gears a little bit, but not too much. Dr. James is an assistant professor at the Benaroya Institute in Seattle. He's an immunologist. He's also been in the field for a while. So Dr. James, tell us what a tetramer is.
Okay, great. So I mean, I'm sure some people on this call will have more knowledge of tetramers than others, but I've tried to make this as inclusive as possible. So my goal here today is really to highlight a special resource that we're trying to build as part of the HIRN resource browser and repository, and that is a listing of tetramers to kind of answer the question for those that are trying to get into these type of experiments for the first time. The very practical question of which isolated tetramer should I use for my experiment? And so what we want is really a vetted list of tetramer reagents that if you order them from the right source and apply them the right way, you have a chance of generating the type of data that you want to generate. So that's the goal. Just kind of facilitating this type of research in the community, which is using techniques that are near and dear to my heart and to a few others that I see on the participant list here. And so just to give a quick overview, so I'll talk just briefly about the development of isolated tetramers, and that kind of folds into kind of which resources are available. I'll talk about what I consider to be some reliable sources for HLA tetramer agents, and if I have omitted your favorite one, I apologize, and you can bring that up during the question and answer period. I'm going to talk just briefly about direct tetramer-based flow cytometry assays that we're using in the lab to generate data, and how those can fold into the application of tetramers to generate omics type data sets, which is something that we're actively doing now. And then I'll yield to the question and answer portion of today's meeting, so that people can ask both me and the other speakers any practical questions that you have about the resources that we've been talking about. And so, on this slide here, shows a diagram of basically how we build an HLA tetramer reagent. This particular example is an HLA class 2 tetramer. And so each tetramer, of course, is built from a monomer. And these monomeric units are actually, dimers of the HLA class 2 alpha and beta chains, which for HLA class 2 are, fairly long molecules that can be highly polymorphic, especially for the DQ and DP molecules. For HLA-DR, the alpha chain is actually dimorphic and tends not to vary. The strategy that we and others have done is linking the alpha and beta chains of HLA class 2 using lysine zippers and adding a single biotin site to allow tetramerization around streptavidin. For HLA class 1, the story is a little bit different, because the beta chain is, in that case, beta 2 microglobulin, which is invariant, but that can still be biotinylated. The strategy there is to usually refold the alpha chain around beta 2 to produce the tetramer agent. But by either approach that you use, where you end up is having hopefully a functional alpha beta dimer, which is usually stabilized by a peptide. If you add the right amount of streptavidin, that can be assembled around streptavidin to give you what's traditionally called a tetramer. In reality, streptavidin actually is slightly self-aggregating, so these really aren't tetramers, they're usually multimers. And so you'll see those terms referenced interchangeably in the literature. But tetramer, multimer, the basic idea is that we want to increase the number of alpha and beta receptors available on this reagent, so you can get a stable, long-lived interaction with the antigen-specific T cells, which labels it in some sense analogously to how an antibody would label a cell of interest. But here we're labeling just TCRs that see our peptide HLA complex of interest. And so then a key question then becomes, well, what epitopes do I use, and where do I get my tetramer reagents from? And here's sources that I personally have used and that I can vouch for their proteins. The NIH tetramer core is, it was founded by Jen Albin, who invented the tetramer during his time at Stanford, and they have continued to operate at Emory University, and they're an excellent source for both class 1 and class 2 reagents. Nice thing with the NIH tetramer core is that you can actually get proteins from them for free, because they have NIH funding to distribute that as a source to the community. And so that's a great option if you want to kind of cut your teeth for the first time on tetramers. They do have some requirements as far as how they prioritize orders, and they want you to use very well vetted HLA peptide combinations, and you need to fill out some paperwork to explain why you're doing the study that you're doing and why you've selected the tetramers that you have used, and they want to make sure that you're not using them for commercial purposes, but a great option. Another source that I can vouch for is the Benaroya tetramer core, which I managed for about 15 years. They exclusively make HLA class 2 tetramer reagents, a very broad range. These are not free, but they're distributed more or less at cost to the research community. So not the same funding structure as the NIH tetramer core. They tend to very rapidly produce reagents, and numerous publications have been put forth into the literature using reagents from the Benaroya tetramer core. Their current management remains very collaborative, and they can make a lot of recommendations as far as which HLAs are available, which peptides have worked for them in the past. If you're willing to pay a little bit more, or if you want to use tetramers for commercial purposes, clinical trial monitoring, or if you happen to be from a company, then these three bottom sources are, maybe your best option. MBL International licensed their technology from Jen Altman. They were formerly known as Beckman Coulter, but they've gone through a name change and change in sponsorship, if you will. But they distribute both HLA class 1 and class 2 tetramers that work, I guess, in my hands, not quite as well as from the NIH and BRI, but still, quite good in most applications. Another key option is Immudex, which distributes HLA dextramers, Those have a slightly higher, I guess, numerical, number of HLA molecules on their surface, and have the flexibility of the dextran backbone. So that's a great tool. And finally, ImmuneAware, which is a company that was founded by Søren Boos in Copenhagen. He was a pioneer in revolutionizing both HLA class 1 reagents and also a series of very unique and interesting binding assays. And they're among the first to supply easily loadable reagents for HLA class 1. So I can vouch for all these sources. The other big question is, okay, how do I choose an HLA type, and how do I choose peptides? And that's basically dictated more by epitope knowledge than anything else. The figure here is from a review article that I co-wrote with Teresa and Roberto and Sally that many of you have probably saw in diabetes. And this just highlights basically the distribution of epitope knowledge. And what I want to highlight is that if you're trying to choose a tetramer, a lot is known about HLA-A2, a lot is known about DR, O401 and DQ8. And there's big gaps for some of the other HLA types that you might be interested in studying for specific cases that you want to look at. And that's an issue that pops up if you are part of the HIRN network or part of NPOD, or there may be a really interesting case that pops up, and then to your dismay and consternation, they may not be DR0401 or DQ8 positive, and then you kind of have to scratch your head and say, okay, well, what tetramers do I need to to select that I can use them? There's also a lamplight effect as far as antigens, and that certain antigens like insulin and GAD are a lot more strongly studied than others. That's something that we know that are working on to kind of extend that. But basically, what I'm trying to do with this research is obviously have A2 and O401 be well represented, but it's a work in progress. But I've been trying to fill in other HLA peptide combinations for which I can vouch for the tetramers, and that maybe give you a little bit broader coverage. As an example of that, I have uploaded a DRB4 tetramer. DRB4 is the HLA class 2 allele that's most widely expressed in human populations, and there's a good insulin tetramer for that one. And so I was sure to include that in the resources. I'll say as an aside that I don't claim to have exclusive knowledge about which tetramers are good, and I know there are others on the call that have done a lot of work, staining tissue with tetramers, for example, or defining HLA peptide combinations that are disease relevant, or tetramers that are useful. And I would love to have you all join me in building up this resource into something that can be the best that it can be. So feel free to either contact me or work directly with the resource browser to get your data embedded into this this case resource as well. But where we've landed so far is that we have the beginnings of what is a special collection. And so if you go to the web page and click on this will link you to the list of HLA class 1 and class 2 tetramers that I've uploaded so far. Again, it's a work in progress, and it'll take me a while to get everything filtered in there that I trust. And again, I invite you all to contribute to this as well. But the hope would be that this will allow people to effectively answer that question of what tetramer should I use, and where can I get it from. And if you go to the browser, what I've attempted to do for each of these is to give fairly accurate information about where these tetramers come from, and they include a link that you can follow to the actual source vendor sites for these different resources. And so I guess if I put up a class 1 tetramer from the NIH tetramer facility, there's a link in there where you can actually go to the NIH tetramer facility webpage and create an account and actually request that tetramer if your heart so desires. And the same being true of the BRI tetramer core and class 2 reagents. So this will get you where you want to go. And so again, now to highlight just one particular application of tetramers, we use these to follow what I would call the numbers problem of enumerating epitope-specific T cells in a blood sample. And that numbers problem is that especially for autoreactive CD4 positive T cells, they're extremely rare. And again, in the millions of PBMCs you get from a drop of blood, there may be as few as 3 to 10 CD4 positive T cells per million that see your epitope of interest. And so, getting that as a convincing staining population requires A, a lot of blood, but B, some technical trips. And in our hand, the technical trick that we use is a magnetic enrichment strategy, where we can, pre-label the cells with the tetramer of interest, and then basically either using a recombinant tag on the HLA, class 2 molecule, and the reagents from the BRI tetramer core actually have a MIC tag on them, or using an anti-fluorophore enrichment strategy. You can basically, you can label your sample with the tetramer of interest. You can add magnetic beads that will stick to the tetramer, either through the MIC tag or through something like anti-PE or anti-APC beads. And you can use Miltenyi or stem cell beads to then enrich your T cells of interest, and you can get easily a 100 to 1,000-fold enrichment of your tetramer positive population, from a couple of dots to a large cloud of tetramer positive T cells. If you save a pre-enriched fraction, you can get a total estimate of your T cell count and actually calculate exact frequencies of those T cells. And this example is flu, but this is real data where, the small sample that you run beforehand really does look like this. The enriched sample really does look like that. And in my hands, at least, that's a whole lot easier to gate. So to me, this makes it tractable to look at tetramer positive T cells, and it gives you a sortable population if you want to do more sophisticated analyses like the ones that I'm about to talk about. And so, one way that we've made this more practical is that, obviously, if you're staining with just one tetramer, it may be, highly positive in some subjects and, mostly negative in others. And so it's highly desirable to multiplex. And so we've spent a lot of work to develop multicolored tetramer staining. What we discovered is that, especially for HLA class 2 tetramers, only certain labels work. And so it turns out that if you want to effectively label and enrich T cells with class 2 tetramers, you have to use the large fluorophores, things like PE, the PE tandems, and APC. But fortunately, that still gives you enough of a tractable option to be able to label either different individual epitopes by doing things like dual encoding, which I'm showing here on the left, or by pooling groups of tetramers from single antigens. You can have tetramer pools 1, 2, and 3 corresponding to antigens 1, 2, and 3. Label those with your large fluorophore of interest, and basically, ask the question of, what's the frequency of T cells that are responding to, maybe, GAD epitopes pulled together, IA2 epitopes pulled together, and insulin epitopes pulled together, and get some relative enumeration of those. And that's something that that we've published on. You can find the paper, so you can ask me about that if you want. But the most exciting thing to do is, rather than just counting those T cells, is to fold those into the emerging omics platforms that are emerging right now. And we actually have this working pretty well now for both HLA class 1 and HLA class 2 tetramer, where you can stain, enrich, if desired, and then either sort those single cells into plate-based assays, or put them into chip-based assays, or even put them onto the 10x platform. And then, either by plate sorting or your cell sorting technique of choice, those cells can be analyzed for gene expression through RNA-seq. You can assemble TCR clonotypes, and you can also collect flow index panels and give you kind of a three-way analysis of those. And this is exactly what we're doing for antigen-specific T cells, from patients during the natural history of disease, and through things like the oral insulin and teplizumab prevention trials, to get an idea of how those T cells may be able to be classified not only by specificity, but how expanded their TCR clone types are, and what their gene expression profiles are, and how they can be bent based on that information and their surface markers into different phenotypic families, if you will. And I think this type of cutting edge analysis is really going to be informative to really kind of winnow down, more than just bean counting of how many antigen specific T cells are, but what the flavors and phenotypes are they, and how much they may be skewed towards inflammatory versus exhausted or regulatory phenotypes at various stages of disease and in response to different therapies. So all this, I think, is worth doing. And so I guess I hope I've sold you on the idea that using tetramers is a cool option and is potentially powerful, and that it's something that it's worth branching out and trying if you feel so inclined and have questions that you want to ask that include antigen specificity as a dimension. And so I'll go ahead and stop there and invite my co-speakers back online to address any questions that people have.
Thank you very much.
Thank you, Eddie. That is terrific. And so if any of you have questions, could you please unmute and ask them, or you can raise your hand to indicate you have a question. Okay. And I really appreciate John, Sarah, and Eddie for highlighting the resources available through the HIRN resource browser. That was a terrific overview. Thank you. Any questions at this time?
Sarah, your ALOX deletions in males and females look pretty similar. So I imagine you don't conclude that there are any sex differences. Am I right?
Not with ALOX 15. We are interested in looking at sex differences between the female and male entities. And there's people that look at that as well. But with ALOX 15, I don't think there will be. And then when we study type 2 and non-NOD specific stuff, then we do use both human and females, females and males, especially in the human islets. So I think it's important to study, but in terms of NOD mice, it's females are way more practical.
Thank you.
Excellent. Any other comments, suggestions? We're always very happy to hear suggestions for additional resources or metadata about the resources that you'd like to see added. So please always keep in touch with us at the HIRC about that.
I see Bhawana, I think, do you want to ask a question?
Yeah, I have a question to Sarah. So when you knock out this ALOX 12, have you ever seen any effect on alpha cells, or like what happens to alpha cell in that deletion?
So
just out of curiosity,
yeah. We don't see any major differences in alpha cells. We did, obviously with single cell RNA sequencing, got alpha cells out, and we see, we didn't see much with the alpha cells. There was a couple genes that were changed, but not many. So I don't think ALOX 15 is expressed in the alpha cells. It's really specific to beta cells.
Yeah, but do you see any transdifferentiation of alpha cells into beta cells in this knockout? Like anything of that sort happens? Or?
There was no suggestion that that was happening. I think we're preserving the beta cells there versus making new beta cells or transdifferentiating alpha to beta cells. Really, with the alpha cells, we didn't see anything in the transcriptomics that would suggest that they were trying to convert or something.
Okay. Thank you.
You're welcome.
Hi, Sarah. This is Jay.
Oh, hi.
Yeah, just to be curious, have you taken a look at the PDL1 expression in female and male mice?
We have not looked at PDL1 expression in the male mice yet. And so that was all of that was done in female mice. But I think that is definitely something that we should do, and something that we're trying to go more. We were just talking about this a couple days ago. More look at the male and female differences. Especially one of the things I didn't mention was one of the other genes that was highly upregulated was estrogen receptor, which actually tends to get activated more in the males. So it's something that we're going to be looking at, but we have not yet.
Okay, thank you.
Great. Any other questions? Okay. Well, if not, I think we'll sign off for today. And thank you all very much for joining. Again, thank you to Sarah, Eddie, John for a great overview. And do keep in touch if there are other resources you'd like to see out there. And we hope you'll all avail yourselves of the library that's in the HIRN resource browser. Thank you and have a great day.